I'll finish my talk, which I'm sure all of you know the name, right? Max Perut and Kenru. Hemoglobin and myoglobin. Is there anybody who hasn't heard the word hemoglobin? Raise your hand. Hemoglobin, you haven't heard. You are a computer science. Huh? Oh, you know. Huh? Okay, life sciences. Hemoglobin is a protein which carries the oxygen in the blood, right? Is there anybody who doesn't know? Myoglobin is a smaller protein which must. Very good. How do we know so well of these two proteins in the human body? Because of these two great scientists, Max Perut and Kenru. They spend their entire life only studying these two molecules. Imagine both got Nobel Prize. So a human body has 26,000 genes, 25,000. And a variation of protein, maybe 500,000. If 500,000 proteins, we have to understand the way Max Perut and Kenru understood the chemistry, the biology, the genetics, we will need 500,000 Nobel laureate. Possible? Not possible. So how do I do that? So the second approach, what we call crowdsourcing. Open source drug discovery is not looking for a single Nobel laureate, looking for a collective Nobel laureate. Tell me, who built Taj Mahal? Raise your hand. Who built Taj Mahal? Huh? Shah Jahan. Who? You know their name? Right? Taj Mahal was funded by Shah Jahan. OSDD is funded by CSIR. Zakir is the project director. I happen to be what is called mentor. Mentor means who knows nothing. He just philosophizes. So you are building Taj Mahal. You are building the new generation drugs discovery platform. And you are one of those thousands and thousands that will come over the decade who will eventually make drug discovery simple. So the chemistry of drug discovery is nothing but understanding ourselves, understanding the way these two molecules. It is observed the people where you have high sickle cell anemia, the people don't die of malaria. The people die of malaria where there is no sickle cell anemia. Now this is a big problem. Now it came out through simple chemistry. When there is a change in the amino acid, in hemoglobin, beta chain, a glutamic acid is replaced by valine, then the cell carries one. When the cell carries both, the cell is sickled and dead. When cell carries 50% of valine containing protein and 50% with glutamic acid, and a malarial parasite enters that cell, when it enters the cell, it reduces the pH, that is, it changes the pH. In changing the pH, only the valine containing cells which has the hemoglobin with valine, aggregates and burst. So only those cells in which malarial parasite has entered dies. So malaria dies. Now you see, here is the disease, how nature has been fooled. It becomes an advantageous. Correct? So therefore, you have to understand the tuberculosis chemistry the same way. As I said, we can't have that many 100,000 Nobel laureates, but if we put thousands and thousands of people, because they did not have internet that time. If Max Perut and Kenru was now today, they would not have worked themselves whole life. They would have solved many, many structures using OSDD model. Okay? So that's the approach. So at the end, I'll show the last slide to tell you, eventually, chemistry of life is nothing but, human body is a chemical factory. How much kilocalorie food you take? 2000. Correct? Somewhere between 1600 to 2200. Okay. For 24 hours. Right? Now tell me, if you convert that, it is about 86 calories per hour, which is like a 60-watt bulb. The PC itself takes 50 watt without the monitor. With that we do wonders, no? So human is the most energy efficient machine, a chemical factory which is carbon optimized, energy optimized, waste nothing. And you have to remember, tuberculosis creates havoc in that machine. So you are handling a big difficult problem. But it is handleable because it's not 5 people trying it. It's 5,000 people trying it. And we are not looking at all together. We are looking piece by piece. So it's like jigsaw puzzle. You have a jigsaw puzzle of 1,000 pieces. I'm sure in this room, if I put a 1,000 piece jigsaw puzzle, in 3 days all of you will finish it. But if I give you alone, you'll take 5 years to do. And that's the challenge. So the idea is, it's an amazing machine. Everything happens here. Information to idea, oxygen to carbon dioxide. And anything that goes wrong, the disease takes place. So you have to now understand not only the tuberculosis organism, which we do, but its interaction with the proteins in the human, and figure it out which activity of tuberculosis is dependent on which interaction, and how can I block that chemical interaction, which is charge-shape complement. And that's all is the chemistry of drug discovery.